Of course, you've probably all heard that we now know that we got HIV from chimpanzees, but when we gave it back to them, they hardly ever get AIDS. That's a good example. There are many probable differences, and there are a bunch of possible differences. Now, there's no way I can cover all these differences in the short period of time here. There's some references down here that those interested might look up. So I'm just going to pick 4 of these, and I'm going to be selfish and pick the examples where there's some relationship to our own work also. But in general, I think it's reasonable to suggest that the major diseases of given species are likely to be related to maladaptations during the recent evolutionary past of that species. After all, absent a new pathogen or a toxin, if things are working well, they're less likely to go wrong. And so it seems reasonable to compare disease incidence and susceptibility between humans and our closest evolutionary relatives. But I'd like to go further based on what I'm going to present, that more research on great ape diseases is needed not because they're useful models of human diseases. We put all these captive apes together as models of human disease, but because they are bad models of many common human diseases. I think that's what's really fascinating. And further studies of great ape diseases will benefit both humans and apes. We shouldn't be using Harrison's textbook of internal medicine for apes. So let's pick one here, malaria. Falciparum malaria, one of the biggest killers worldwide. And it turns out that humans and chimpanzees have distinct malarial parasites. In experiments that will never be repeated in the Belgian Congo in the 1920s, the observation was made that chimpanzees and gorillas had on their blood smears a parasite that looked just like Falciparum malaria. So the story was the mosquitoes were biting the chimps and then biting the humans. So these Belgian scientists took infected chimps and infected humans and cross-transfused them into, quote, normal humans and normal chimp volunteers. And a terrible experiment, but the result was very clear that there was a different parasite. Humans didn't get sick with this chimp parasite, and chimpanzees had mild or no infection with the big human parasite. So, malarial parasite life cycle is very complicated, but just keep it simple. Mosquito bites you. A lot of stuff goes on in the mosquito. Mosquito bites you again. It gets into your liver, divides, and then gets into your red cells, and eventually gets back out again. And this red cell stage is what we were interested in, because when we started this whole project, the way we had found the difference between humans and chimpanzees was to look at red blood cells, and the sialic acids were completely different. And it turns out that when the parasite invades the red blood cell, it uses sialic acid as a handle to get hold of. So it turns out that human and chimpanzee parasites have different sialic acid binding preferences. And just as we had guessed, the human parasite prefers to bind the human sialic acid, and the chimp parasite prefers to bind the chimp sialic acid. So in part of this publication, we suggested that maybe ancestral hominins escaped the pre-existing Reichnoi when they lost GC expression. In fact, maybe that was the original selection, maybe, who knows, for having lost GC. And that Falciparum then evolved from a strain of Reichnoi which took advantage of the fact that there was an excess of AC. And we said that also because there's some evidence that the Falciparum malaria evolved very recently. And I'm very happy to report, those of you who came to the last symposium heard a little bit of this from Francisco Ayala at Irvine, that he and his colleagues have found strong evidence for this. This is a big family tree of all the different types of malarias, and here is Falciparum malaria. And Falciparum malaria belongs to a very small group of parasites that are related to one of the many isolates of Reichnoi that Ayala's group isolated. So it appears that back here we may have escaped the big ape malaria. And eventually, as always happens with pathogens that always win, the pathogens one strain figured out how to bind AC. At least that's our hypothesis. So it's consistent with what is known. Okay, epithelial cancers. One of the surprising to me, I was once trained as an oncologist, is cancers of the head and neck, stomach, colon, prostate, uterus, lung, pancreas, etc., have never been reported in great apes. In fact, they're exceedingly rare, to the point that if there was a case, they tell me it'd be a reportable case. Now, of course, these are cancers of older age, and as Tuck Finch pointed out, there's age issues, but still by now you should have seen a few breast and colon cancers at least. So there's many theories about this, and I just want to tell you our theory that might contribute to this. And this is from the World Cancer Research Fund, the top 10 things to do to avoid cancer. And one of them is to limit intake of red meat. And there's very clear epidemiological evidence that intake of red meat and, in some cancers, milk is clearly associated with a 2 to 3-fold increase in cancer with much lower incidence in groups that don't consume much of this. And so we just had this paper out on a human-specific mechanism for antibody and diet-mediated carcinoma progression. And I'm not going to show you the data. I'm just going to summarize what we think we found. And that is, we had a common ancestor. We lost GC. And then we domesticated a lot of animals that are full of GC. And in modern times especially, we've been eating more and more of this GC. And we've shown by human experimentation and by studies that Nissi Varki has looked at tissues in humans that even though we shouldn't have any GC in our body, it is incorporated into the very places where these cancers appear. And meanwhile, we found that all humans have antibodies against GC. So we have a peculiar situation where a foreign molecule is like a Trojan horse. It's biochemically not foreign, it's immunologically foreign. So we have this ongoing antigen-antibody reaction going on, which would generate, in the lining of the epithelium of hollow organs, we believe, chronic inflammation. And as you've already heard, chronic inflammation is a great way to facilitate the progression of cancers. And we have mouse data in this paper, if you're interested, that really supports this idea that, in fact, this combination is facilitating the progression of cancers. Of course, a lot more epidemiological and other data is going to be needed before we can really say that this is for sure. But it's completely consistent with everything we know. And, of course, carcinomas being rare in apes. Myocardial infarction, heart attacks, the single biggest killer of westernized humans. It's basically a blockage of a blood vessel to your heart, coronary artery, and you die or you get very sick or die because you don't get the blood supply. So based on a lot of work involving many people at primate centers and Nissi Varki and myself and others, we recently had a paper that is now in press in a new journal called Evolutionary Applications involving us and the Yerkes Primate Center and the Primate Foundation of Arizona, which is entitled Heart Disease is Common in Humans and Chimpanzees, but is caused by different pathological processes. So when I first went to the Yerkes Primate Center, I said, well, what's the commonest cause of death in chimpanzees? I said, well, heart disease. I said, oh, it's just like humans, nothing different. And indeed, if you look in this table that was data put together by some of these people, if you look in the recent times, heart disease is a major killer of chimpanzees. And this is also true of gorillas and orangutans. But the disease, it turns out, is completely different. And this is work by Nissi Varki and Dan Anderson, and these are sections of the heart. And this is just a straight section. And what you see here is a chimpanzee who died of a sudden death. And you see all this white stuff in there. So in other words, the heart muscle is kind of missing. And you stain it with this trichrome stain, which stains fibrotic lesions, and you see this massive amount of this blue stuff there. So what is happening to these animals is they're getting this massive fibrosis throughout their myocardium, and the heart attacks they're getting has nothing to do with blocking of a coronary artery. It's a sudden arrhythmia. Their rhythm goes wrong, and they drop dead. Or they die of heart failure, and their hearts get big, but it's not because the blood supply is low. So just to be clear, it's not that there's no atherosclerosis at all in chimps. It definitely occurs, and they do get a few strokes and so on. But the severity is nowhere near what you see in humans. So in humans, here's a typical atherosclerotic plaque full of cholesterol. This is what would eventually expand and block this artery. Here's a chimpanzee coronary artery. And so we think there's a very distinct difference. And there's this fibrosis going on. Now, Nissi said, well, is this something that's happening only in these sick animals? So she decided to do the same stain on normal hearts of relatively young animals and humans. And what you can see here, it may be a little bit hard to see, humans do have this fibrotic tissue appropriately along the blood vessels and in the major blood vessels. But you see in the chimpanzee, gorilla, and orangutan, there are these lines of this stuff. The chimpanzee heart seems to be divided into sections by these fibrotic planes. And maybe the disease is some sort of aggravation of this thing. So we really don't know what's going on in terms of understanding this process. But there are two mysteries to be solved. Why do humans not often suffer from this disease that's so common in our closest evolutionary cousins? This is not at all a common disease in humans. And why do great apes not often have the kind of heart disease severity that's so common in humans? And we have data which we're working on right now. And by the way, they have actually higher cholesterol levels than we have. They have the ApoE4 allele, although you've heard that might be not entirely clear what that means. Higher LPA levels, sedentary lifestyle, hypertension, and so on. So actually, we have some data for the same kind of phenomenon. GC is being incorporated into large vessel endothelium, chronic inflammation, and we think that's exacerbating heart attacks. And cholesterol is the key. So I think I'm going to skip the next two slides. It basically shows GC in the blood vessels of humans. And this is work in which actually human antibodies have been taken from human serum and shown to bind back to the GC on the blood vessels of humans. And there's in vitro work that supports this. So finally, a quick one going back to preeclampsia, the disease that Kurt mentioned, this apparently uniquely human disease. I'm going to skip these two slides, which is basically duplicative of what Kurt told you about this disease. And what Virginia Wynn and her colleagues at Colorado have been looking at this. But we, meanwhile, found by serendipity that human placenta, the trophoblast, has a lot of this molecule called Siglex-6 that binds sialic acid. And this is not true in the apes. But the gene is present in humans and apes, but found on B cells, but this is uniquely human. So Virginia Wynn and Susan Fisher and colleagues, they did a microarray analysis, say what genes are being expressed in the placenta. And they found the usual culprits known in this disease, CRH, VEGF, leptin, and they found Siglex-6. So Siglex-6 is markedly upregulated in preeclampsia. And it turns out we had earlier shown that Siglex-6 binds leptin. So that's a known interaction. We have no idea what this means, but maybe this is some connection to why humans might get this strange disease. So finally then, I'm very happy to report that after repeatedly pestering the curriculum committee here, for the last 3 years, I've been giving lectures on human evolution, the basics and relevance to human health and disease. And in fact, they very grudgingly gave me 2 hours to do this, but I got the best response I've ever got for any lectures I've ever given to any students. And so they had to let me continue to lecture. And I'm trying to convince them that the first thing a medical student needs to know when you come in the door is what is a human and where did it come from? And I might succeed in that. So we'll see. So this is the list, and we can talk some more about it later if you like.